Structure of DNA. Objective: to learn about the general structure of deoxyribonucleic acid. DNA, deoxyribonucleic acid, is present in the nucleus of every cell. It is present as a highly coiled structure in the form of chromosomes. In 1953, James Watson and Francis Crick proposed the structure of DNA. DNA is a macromolecule polymer made of subunits called nucleotides. The nucleotides are made of 3 units: a phosphate group, a 5-carbon sugar called deoxyribose, and 1 nitrogen-containing base. The nucleotides are arranged into chains, or strands. Each strand has a backbone made of alternating groups of deoxyribose sugar and phosphate groups. The sugar molecule of one nucleotide is linked with the phosphate of the next nucleotide by a phosphodiester bond. There are 4 nitrogenous bases in DNA: adenine and guanine, called purines, and cytosine and thymine, called pyrimidines. The bases are attached to the sugar molecule. 2 strands of nucleotides pair together in a specific way. Adenine pairs with thymine, and guanine pairs with cytosine. 3 hydrogen bonds are present between C and G, while 2 hydrogen bonds are present between A and T. AT and GC base pairs are said to be complementary. A DNA strand is assigned a direction based on what group is found at the end of the strands. If one end has a free phosphate attached to the 5th carbon of the sugar, then that end is called the 5' end, or 5' end. If the other end of the strand has a free hydroxyl group attached to the 3rd carbon of the sugar, then it is the 3' end, or 3' end. The 2 strands of DNA are anti-parallel to one another. One strand is 5' to 3', and the other is 3' to 5'. The paired DNA strands form a double helix, which looks like a twisted ladder. DNA contains the instructions needed for an organism to develop, survive, and reproduce. Genes carry biological information that is copied and transmitted to the next generation. Each time a cell divides to form 2 daughter cells. A stretch of DNA, or DNA sequence, contains instructions to make a protein and is known as a gene. In humans, the size of a gene varies greatly between 1,000 and 1 million bases. Summary: inside each cell lies a nucleus that contains long strands of deoxyribonucleic acid. The double helix structure of DNA is made of phosphate, deoxyribose sugar, and 4 nitrogenous bases. The strands of DNA are anti-parallel in arrangement. DNA encodes genetic information.